Now it's my pleasure to introduce today's speakers, Drs. Kostas Bourtzis and Jeremy Bouyer, who are both from the joint program of the United Nations Food and Agriculture Organization and the International Atomic Energy Agency, focused on nuclear techniques in food and agriculture. This program has been a major force in the developing and delivering SIT solutions to UN member states. Kostas leads the Molecular Biology and Genetics Group at the IAEA and has done so since about 2013. Before joining the IAEA, Kostas's research focused on his research at the University of Patras was on the study of interactions between insect pests, disease vectors, and symbiotic bacteria, with special emphasis on Wolbachia-mediated cytoplasmic incompatibility, genetic manipulation of Wolbachia, molecular mechanisms of incompatibility, and Wolbachia genomics. Kostas has made important contributions to the efforts to use Wolbachia-induced cytoplasmic incompatibility as an insect population suppression strategy, in addition to his contributions to the SIT efforts during his years at the IAEA. Jeremy is an entomologist and vector biologist at the IAEA and is also a researcher at the French Agricultural Research Center for International Development in Montpellier. His expertise is on vectors, including mosquitoes, tsetse flies, and has a broad interest in controlling invasive insect species. He's the principal investigator of a project called Revolink, which is developing novel ways to expand the utility and enhance the effectiveness of the sterile insect technique. With that, let me say welcome, Kostas and Jeremy. Thank you for your willingness to participate today. And Kostas, let me invite you to share your presentation and let me know when you bring it up. Great.
Yes.
I can see it. You're in presentation mode, and
okay.
The floor is yours. Thanks.
Thank you, Dave. Good evening from Vienna, or good morning there. First of all, I would like to thank Dave and his colleagues for inviting us to give this webinar. And what Jeremy and I would like to do over the next 45-50 minutes is to present you the work that we have been doing in the frame of the joint FAO-IAEA insect pest control subprogram in respect to the development and application of SIT against major mosquito vector species such as Aedes aegypti and Aedes albopictus. And why is this important? It is important because, as you know, these mosquito species are major vectors of human pathogens such as dengue, chikungunya, and Zika. And according to WHO, CDC, and ECDC, just in the year of 2016, there were more than 3 million cases of dengue, more than 1.2 million cases of chikungunya, and more than 1.5 million cases of Zika. And unfortunately, the problem is huge, and the currently available tools to address the transmission of these pathogens and the disease they cause are limited. There are no efficient drugs or vaccines, and the only tool that we have is to control the vector species, the vector mosquito species, and this is currently being done mainly by sanitation or the use of insecticides. But neither of these tools is efficient and sustainable, and let alone the fact that insecticides, the excessive use of insecticides have triggered the evolution of insecticide resistance, and in addition to the major threats they pose on the environment and human health. So what are the alternatives? The alternatives is, novel and environment-friendly and species-specific vector control methods, such as the Sterile Insect Technique. Our sub-program has developed a video, and I would like to kindly ask Dave to present this, which shows very briefly the principle and the major components of the SIT package.
Mosquitoes. The battle continues, but the battlefield is changing. Throughout the millennia, people have fought mosquitoes and the diseases they transmit. Now, with globalization and with climate change, mosquitoes and diseases are spreading. Mosquitoes are becoming resistant to insecticides. The diseases they carry are becoming resistant to drugs. And mass use of pesticides required for control has negative effects on public health and on the environment. But there is another battle plan, the Sterile Insect Technique, SIT. SIT is a type of birth control for insects. Already successful in controlling major crop and livestock insect pests, SIT is being refined and validated to control mosquitoes. Special factories rear millions of male mosquitoes, sterilize them with radiation, and then release them into target areas at regular intervals. They mate with the wild females, but the males are sterile. There are no offspring. When sufficient sterile male mosquitoes are released, each successive generation will have fewer wild mosquitoes, and the population will be suppressed. Success will depend on people in villages, towns, and also cities, collaborating with public campaigns to eliminate mosquito breeding sites in combination with traditional control methods and the release of sterile males. This integrated management reduces the mosquito population enough to prevent disease transmission and nuisance biting. As there is no environmental pollution, no development of resistance, it is sustainable. SIT offers an additional method to control mosquitoes and the diseases they carry, and thus to reduce human suffering in many parts of the globe.
Thank you, Dave. So as mentioned by Dave in his introduction, SIT has a long history. It was initially conceived by Ed Nippling in 30s, and it was applied for first time against the New World screwworm in the 50s. And it was very successful in eradicating this veterinary pest, this livestock pest from southern US states and the entire Central America. And during the last 60 or 65 years, it has been developed and successfully applied against several plant pests, including fruit flies and moths, as well as against additional livestock pests, such as tsetse flies. And very recently, we have been developing the SIT package, and we are testing it against major mosquito vector species, such as the Aedes aegypti and Aedes albopictus. There are 4 strategic options that one can use the SIT and apply the SIT. The first one is called suppression, and in respect to the vector species, the goal there is to reduce the population below the threshold required for disease transmission. Another way is to use it for containment, and this is to avoid the spread of introduced non-native vector species or pest species, or one can use it for prevention, in order to avoid establishment of non-native vector species or pests. And also another way is to locally eradicate a population of a vector species in order to develop areas free of it. So no matter which strategic option one decides to use and apply SIT for, there is a major step in this whole process, and this is the sex separation step. It is particularly important for vector species because in addition to the improvement and enhancement of the cost efficiency, the cost effectiveness and efficiency of the SIT application, for the vector species it's important to eliminate females because the females bite, blood feed, they transmit the pathogens. And then if we remove the females, then one can only sterilize by radiation and release males in an area. How this can be done, there are several ways, but it has been shown that the genetic sex in strains is perhaps the best option. In order to develop genetic sex in strain, we need two components. The first one is a selectable marker, which is usually a phenotypic trait, and then we need to use this marker to sex. There are several mutants for both Aedes aegypti and Aedes albopictus, which are based on color. There are eye color mutants and larval body color mutants, and some of them are shown in this slide. And we have decided to use the eye color mutants in order to develop a genetic sex in strain which can support the SIT application against Aedes aegypti. And we took advantage of the fact that both the red eye and the white eye mutations had been found to link to be located on chromosome 1 and linked to the male determining region. So what we did is to do essentially Mendelian genetics. We crossed a wild type male with a mutant female, red eye or white eye, and then we took the F1 males and we back-crossed them with the parental mutant females, the red eye and white eye respectively. And then in the F2 generations, we had males which were having black eyes and females which were having either red or a white eye eyes. You know, one can continue inbreeding this line, and then it can always get wild type males and mutant females in the absence of recombination. But recombination exists, and in the case of Aedes aegypti, there is recombination both in females and in the males. In the case of the red-eye GSS, we estimated that the recombinant progeny were in the range of 2 to 3%, while the recombination rate was a little bit higher in the case of the white-eye GSS, in the range between 8 to 10%. However, you know, recombinants pose a major threat in the genetic integrity and the genetic stability of a strain. So it's important, you know, to remove them in order to maintain the integrity and the stability of the strain. So we decided to establish a filtering system and to exploit different threats in Aedes aegypti. So we decided to establish a two-level filtering, one at the pupal stage and the second one at the adult stage. So in the pupal stage, the first step was to sex sort according to the pupal size dimorphism. We know that in Aedes species, female pupa are larger in size compared to the male pupae. And then under the scope, you know, we did two kind of screening. First of all, of course, based on the eye color, selecting only wild type black eye males and only red eye or white eye females, and then confirming, you know, their sex status, male or female, by inspecting the genital lobe at the pupal stage and the antenna type at the adult stage. There is, of course, another way of filtering out and removing and eliminating recombinants, and this is by inversions. We know that chromosomal inversions are recombination suppressors, and we also know that they are naturally occurring in insect populations, including Aedes aegypti. So what we did was to place these red eye and white eye mutations in different genomic backgrounds, using populations, natural populations and laboratory colonies that they were available to us, and they are shown in this table. And but unfortunately, despite the excessive screening that we did, we were unable to isolate an inversion which could reduce the recombination frequency between the red eye or white eye locus and the male determining region. So next we decided, you know, to use irradiation in order to induce an inversion. And we did that using the schemes which is shown on the right side of this slide. So we used wild type males, we irradiated wild type males from the Brazilian natural population. And then we crossed these irradiated males with red eye or white eye females. And then we en masse, this process was done en masse. And then we selected individual F1 males, wild type males, and we backcross them with the red eye females. So we established about 197 male lines. About 70% of them survived, and 1/10 of them, only 7, showed some promising evidence of reducing recombination frequency between the selectable marker and the male determining region. And 2 of them were verified as a good recombination suppressors, the so-called inversion 35 and inversion 5. One of them, inversion 35, was able to reduce 10 times the recombination frequency between the red eye locus and the M locus. So this means that recombination rate was reduced from 2 to 3% to 0.2 or to 0.3%. Which was good news, because this, was next integrated into our genetic section strain. And we established then 2 types of, red eye genetic section strain, one with and the other without the inversion 35. So what we did next was to assess the quality of the developed genetic section strains, the red eye and the white eye GSS, in comparison with the wild type strain, in that case, the Brazilian population. And we assessed the genetic stability of these strains and also their rearing efficiency and their overall biological quality. So with green color, we indicate that this characteristic, this trait was better than the wild type strain, and the red, of course, it was worse than the control strain. So as you see in this slide, the red eye GSS in respect to the genetic stability was much better than the white eye GSS with filtering or without the filtering and removal of the recombinants. In respect to the productivity, fecundity, and fertility, it was similar to the wild type strain, and it was much better. In the white eye GSS was worse in respect to the productivity, but was slightly better in respect to the fecundity. And in respect to the lifespan and flight ability, with or without irradiation of the males, we noticed that the red eye GSS was much better than the white eye GSS, but also it was better, to our surprise, than the wild type Brazilian strain. And in respect to the male mating competitiveness, after irradiating males at the 90 gray, which is the dose that can induce more than 99% sterility, we didn't notice any effect of that irradiation dose on male mating competitiveness. Then, so which was good news. It was, all these data suggest, that red eye GSS could be a promising GSS and can be used, for SIT applications. But we wanted to assess also the role of the inversion 35 after its integration into this GSS strain. So we compared the 2 red eye GSS with and without the inversion. And of course, the one with the inversion is much more genetically stable than the one without it. But this comes to a slight cost in the productivity, and a slight cost in the flight ability, which is used as a proxy for the biological quality of the males. And despite this, slight reduction in the flight ability of the red eye GSS with the inversion 35, I have to note here that it is still much better than the wild type strain. And this was also observed, as I will show later, under mass rearing conditions. Having these 2 versions of the red eye GSS with and without the inversion, we wanted to see how this strain behaves over time in respect to its genetic stability. And what you see in this graph is the percentage of recombinants on the y-axis versus time. 15 generations are depicted in this graph. And as you can see, if we don't include filtering, the red eye GSS without the inversion starts very quickly accumulating recombinants. With filtering, the recombination frequency is maintained in the range of 2 to 3%. However, the strain with the inversion 35, exhibits extremely low number of recombinants but even without filtering. So in the range of 0.2, 0.1, 0.2, 0.3%, even after 15 generations without filtering, this strain does not accumulate, recombinants, which is very impressive and which really clearly indicates the genetic stability of this strain. So what we did next was to test this red-eyed GSS with version 35 under stress conditions, under mass rearing conditions. And to make the long story short, we noticed that the production parameters, the rearing efficiency, in other words, of this GSS strain is very similar to the wild-type strain. In this case, we used the Mexican natural population. The genetic stability is not affected, but what is really impressive and shown in this graph is the flight ability of the GSS with inversion 35, mosquitoes. So you see here the escape rate and the two strains, the wild-type strain and the strain with the inversion. So we applied this flight tube test, which was developed by Jeremy and his group, and which will be explained later by Jeremy. And we saw that the mosquitoes of the genetic section strain with the inversion 35 are much better flyers than the wild-type strain. And again, flight ability is used as a proxy to indicate the biological quality of these males in respect to their longevity, and is directly associated and correlated with the longevity and the male mating competitiveness. So all of this is, taken together, good news, and so that the two versions of the red-eyed GSS with and without the inversion are promising strains that can be used for SIT applications. So we decided to see if they can suppress a target population under laboratory conditions. So what we did was to set up laboratory cages in triplicate, and then release their equal numbers of wild-type males and wild-type females. And then we released 10 times excess irradiated males of these two strains, which have been sterilized by ionizing radiation at 9 degrees. On the y-axis, we show the egg hatch rate, and on the x-axis, you see the number of weeks. And as you see on the left side, the males of the red-eyed GSS, irradiated males of the red-eyed GSS, were able to suppress the target population within 6 weeks, while the males of the red-eyed GSS with inversion 35 required a little bit longer time in order to achieve the same result, to completely suppress the target population in 9 weeks' time, which are, both of them very encouraging results, which gave us confidence that these strains can be used, in small-scale field trials and validate their capacity to suppress the target populations in the field. However, there is, as you know, there is skepticism and in several cases major concerns in implementing mosquito population suppression programs using strains from different regions or countries. So we wanted to address these concerns, and we decided to transfer the sexing properties of the red-eyed GSS with and without inversion 35 in different genomic backgrounds, and see if the sexing properties, are maintained and the strains are genetically stable. So we transfer the red-eyed GSS and the version 35 in the Sri Lanka population, Brazilian population, Mexican population, Indonesia, Thailand, La Reunion, Singapore, and Pakistan. And in all cases, the sexing properties were not affected, and the genetic stability and integrity of the genetic sexing strains was not affected either. And so this means that we have created red-eyed GSS that can address these biosafety concerns. And in addition to that, these strains will have higher probability of success because they will not face any mating compatibility problems. What we wanted to do next is to see if we can, this sexing system that we have developed, at the lab scale can be automated. And the first step to do this is to see if we can have a system which can automatically identify the eye color. So our collaborators established an algorithm which works very nicely and allows successful sexing. And this is based on the fact that if someone uses a powerful light in the infrared spectrum, then the melanized eye of the Aedes aegypti mosquito pupa is seen as a dark binary large object that can be visualized, distinguished, from the visualized and distinguished from the rest of the body. So, and this is what you see here. This algorithm was applied, and tested, and it has shown with a very high efficiency its ability to recognize the black eye of the males. And in the case of the females, because they lack melanin, this appears as a transparent, the area around the eye, and and this is how we recognize the females. So this algorithm, as I said, works very nicely and with 100% efficiency. And we believe now that we have several tools in our hands which can be combined and can be automated, and this is, the next step. So we thought in the beginning, that we can combine the pupil size dimorphism with the eye color sorting at the pupil stage. And as I said, using the red dye GSS with the inversion, the number of recombinants, is reduced to 0.2, 0.3%. So if we apply also a pupil size, filtering system, a sorting system, and these give escapers, in the range of 0.1 or 0.1%, then the combination of these two will result in only 2 females, contaminants out of 1 million or 10 million, females. Very highly close to 100% efficiency of eliminating females and achieving male-only releases. And these two filtering systems and sorting system is compatible with other, mass rearing properties. For example, the male protandry. We know that in mosquitoes, males develop faster, and they pupate and also emerge, much faster than the females. So combining these properties, and engineering and automating the system can allow us, to have a very efficient sex separation system, which can be used for SIT applications in the field. So in summarizing this first part of our presentation, we can say that we have developed two genetic sexing strains for Aedes aegypti, a red eye GSS and white eye GSS, in which males are having black eyes and females red or white. All quality control tests have shown that the red eye GSS is much better than the white eye GSS, both in the genetic stability and in the rearing efficiency. We have developed two versions of the red eye GSS, one with and the other without the inversion 35. Inversion really dramatically suppresses recombination efficiency, and both of them have been used in laboratory cage tests to suppress a target population within 6 to 9 weeks. We have introgressed the red eye GSS with and without the version in 8 different genomic backgrounds. In all cases, their genetic properties were maintained, and these are ready to be tested in the field. And we have also developed an algorithm which allows us very efficiently to identify the eye color of males and females. And this can be used, for automation of the sex separation system, which is the next and challenging step for us. And Jeremy, now is your part.
Thank you, Kostas. I will try to give some examples of operational research that was conducted in our lab to try and help the field programs. So the research is really practical, and our main goal is to help with the field trials. And I will start with the mass rearing component. And the main goal for the mass rearing component is to reduce the cost to produce these sterile males. Because as you know, in fruit flies, they are able to produce sterile males at a very low cost, and the cost of producing these sterile males has been decreased by more than 99% if you compare to the beginning of the programs. So we try to give you some examples first for the larval stage. As you know, mosquitoes have their larvae in water, which is a big challenge because in other insects where sterile insect technique was developed previously, none of them were reared in water. And so we had several challenges caused by water, and the weight is one of them for the racks, for example. But now I will try to give you an example of research that we conducted to reduce the cost, because the reference diet that we used at IAEA before, and that was recommended to member states, was based on a combination of bovine liver powder, Bouyer yeast, and tuna meal. And the problem is that the bovine liver powder is quite costly, up to 70 euros per kilogram. And also, it's not always possible to import it in all member states. So we have problems with projects that could not procure the BLP. So we decided to try several new proteins, and among them, the most promising were insect proteins. So we tried different insects, including yellow mealworm, black soldier fly, and house flies. And at a small scale, the most promising was black soldier fly. And it's an insect that is already used to feed many different animals, including poultry and swine. And it has a very high protein levels and also interesting macro and micronutrients. And we wanted to test it at different scales. So we tried to feed batches of mosquitoes of the two species, Aedes aegypti and Aedes albopictus, and compare the results on production parameters and quality control parameters between our reference diet and this black soldier fly. And we observed that for each of the species, we were able to find a diet combination that was giving similar, if not better, production and quality. And in the case of Aedes aegypti, the diet is a mixture of tuna meal, black soldier fly, and Bouyer yeast. Whereas in the case of Aedes albopictus, the best one was simply a mixture between tuna meal and black soldier fly. So with this diet, we obtain similar pupation rates, production rates, and similar quality with this slide test that I will present later. And we were very happy because we started to generalize its use, and now we have several member states that have been trying to use it. For example, Spain, which has a pilot program, and they are also very happy with it. To give you another example of cost reduction in mass rearing, I will give you an example on adults. So we have been developing mass rearing cages for now 3 years. And the goal was to develop cages that can be built locally by member states and to share the plans on internet so that anyone can produce the cages. So we produce large cages of 90 by 90 by 20 centimeters that can host 16,000 mosquitoes, including 12,000 females and 4,000 males. And we tried to see how they compare to a reference cage, which was iron-based and was quite costly to produce, around 2,000 euros per cage. And we succeeded to develop a cage which is producing the same number of eggs in all mosquito species. And here you have the results for Anopheles arabiensis and Aedes albopictus. All these results can be found in the corresponding papers, and I will be able to share it with you if you need more details. So the last step that was developed recently is that we created a cage with an increased biosecurity, and with the new system, there is no possibility of escape of the females when you collect the eggs. And also it's stackable, and it reduces workload in comparison to the previous one, because only one operator can do all the processes. And the results for this cage, for this new cage, are very promising. They are still not published, but they will come out soon. And the good thing with this cage is that its cost is around 250 euros per cage, which is almost 10 times lower than the cost of the reference cage. Now I will give you some examples of operational research that we did to optimize mosquito sterilization and also to control the quality of these sterile males. So we observed that in different countries that we are trying to produce large numbers of sterile males, in particular in China, we had levels of induced sterility that were below the targets with the doses that were used. And we made the hypothesis that it was caused by an hypoxic environment that is causing some protection against radiations. And so we developed a protocol where we tried to see how the pupae are using the dissolved oxygen in the water. And then we tried to see if we could confirm that we see difference in induced sterility between the batches that are irradiated in hypoxia or in normoxia. So we were very surprised to see when we looked at the consumption of oxygen in closed vials where mosquitoes were put, that the different mosquito species don't react the same. So on the right here, you have Anopheles arabiensis, and it's a species that generally live in a large breeding site. And when it's put in a closed environment, it uses all the oxygen very quickly and die quickly. So they are reducing the amount of oxygen below 0.1% in under 10 minutes. And if you look at the two other main species, Albopictus does the same in 20-25 minutes, and it takes up to 40 minutes for Aedes aegypti to do the same. And the problem is that the irradiation processes are generally taking between 10 and 30 minutes. So we realize that when we do so, we have pupae that are at different levels of hypoxia, all the more that the one that will be at the top of the canister will not be exposed to the same level of anoxia than the one that will be in the middle. And we also confirm that this is creating a large difference in radiosensitivity. And we could see that the mosquitoes that were irradiated in normoxic condition, in red in these graphs, were much more sterile than the one irradiated in anoxia, which confirmed our hypothesis. The problem is that we already knew that pupae are have also different sensitivities to radiation depending on their age. And since it's quite difficult to standardize the production of pupae of exactly the same age, we have a second difficulty with this anoxia, which make it unrealistic to think that we will be able to irradiate huge numbers of mosquitoes without any variability. So we are now shifting to irradiating adults. So in this box on my screen, you can see a carton box with cells of 3 per 3 centimeters. And in this box, you have 35,000 mosquitoes that are maintained at 10 degrees. And we have started to shift to irradiating these chilled adults. And we observed excellent results with a much better quality for the same irradiation and sterility level. And it's mainly because anoxia in itself was reducing the quality of the sterile males coming from irradiated pupae. And this was possible because we used this flight test that was also developed in the last years. And it's a flight test that can give you an idea of the quality of the males. So we validated it against other quality control indicators like survival or mating propensity. And when you want to use this flight test, you put 100 males at the bottom of these small tubes, and the mosquitoes have to fly vertically like helicopters to escape to these tubes. And we observed that their ability to do so with tubes that are around 20 centimeters of height was directly correlated to their quality and probably their competitiveness, because now we have several countries that have adopted the test, and we are trying to compare the competitiveness of the sterile males to their flight ability. We even saw that the flight ability is probably more sensitive than the competitiveness that you can measure in cage or semi-field conditions. So, of course, this flight test is very useful. You can use it at all levels of the production of the sterile males, because these males are mass-reared, they are handled, chilled, transported, released by air, so they suffer different stresses, and it was important to be able to check these different, the impact of these different stressors. And Kostas also showed that it allowed to compare the different genetic sexing strains that can be developed and see if we have males with a good quality. And recently, we also adapted it to Anopheles arabiensis, the vector of malaria. And of course, it can be used for any genetic control measure. And if you look at these graphs, the two on the top are looking at the impact of irradiation on the escape rate. And it's very clear that if you increase the irradiation, you will decrease the quality of your males. So there is a trade-off where you will have sterility upon 99% and still have males that will be competitive. And it's the same for temperature. If you decrease the temperature too much, the males will suffer and their quality will decrease. And also the same with compaction. And that's how we saw that we cannot have a pile of mosquitoes that will be higher than 3 centimeters. And that's why we developed these carton boxes with walls every 3 centimeters. Now I will try to present you some results on how we try to improve the aerial release of sterile male mosquitoes. We developed an automatic release system that was tested in Brazil. And I took this photo so that you can see that the drone that we used in Brazil was a 12 kilograms drone. So it was quite dangerous to fly it over people. So we use it in a rural setting, but in urban settings, of course, it will need to be improved. So we did an experiment in Brazil where we compared two different things. For example, first we compared ground releases in one point in a football field to releases at 50 meters high and 100 meters wide, so point releases. And also we looked at the pattern of the distribution of mosquitoes that we could obtain with release lines that were spaced by 80 meters. So we could see that with these release lines every 80 meters, we could succeed to positive almost all the traps on the ground, and that the ones that were negative were also traps where we did not catch a lot of wild males or even none. And if you look at the correlation between the recaptures of sterile males and wild males, it was excellent. So it showed that we could impose a homogeneous ratio of sterile to wild males over the release area with this drone release system. When comparing the point releases, we saw that the higher you release your mosquitoes, so on the left here you have 100 meters high, in the middle 50 meters high, and on the right ground release. And you can see that the survival is decreasing with the altitude, but the dispersal is increasing. So we will have to find the best threshold in operational programs to get the better coverage while not reducing the survival of our sterile males. And what was really surprising is that during this trial, we only did 3 releases of sterile males, and we already succeeded to impose a 50% reduction of the fertility of the eggs. And we had a ratio of sterile to wild males that was less than 1. So it means that the sterile males had a very good competitiveness, which was estimated around 0.26. So now we are working within the ERC project that was mentioned earlier, which is the Recombinant project, to reduce this drone. And we have one that is now less than 900 grams. And we have tested it at the IPCL already, and we obtained excellent results with no reduction of the flight rate of the mosquitoes that pass through. And now it is being tested in the field in different countries. And the good thing is that you can fly it over urban areas. So now I will speak a bit about what happens when you release the sterile males in the wild. You know that they have to compete with the wild males to mate with the females. And this is measured with what is called competitiveness, which is the odds of a wild female being mated with the sterile males as compared to a wild male when it's exposed to both in equal numbers. And Fried developed an index in the 70s, which is called the Fried index now, and which is based on the measure of the natural fertility of a control population, the observed fertility under a given ratio, and using all these numbers, you calculate what is called the competitiveness. A very important thing is that when you try to do so, in a release area. So here I symbolize the release area in blue, and around you have a buffer of the same size, which is 200 meters. And if you consider a mean dispersal distance of 100 meters for Aedes females, which is common, then you will still get approximately between 1 and 5% of females immigrating from outside in your release area. And the important thing is that if you increase the ratio of your sterile males too much in your release area, then you will have a false idea of your competitiveness. Here on the y-axis, you have the competitiveness, and here on the x-axis, you have the ratio. And we saw that you should keep this ratio between 5 if you want to measure the competitiveness well, and in the presence of invasion from fertile females from the outside. And it is true for all genetic control techniques. So we tried to review what was published in the literature, because we observed that many genetic control programs started their papers by saying that it's not possible to produce competitive sterile males. And we saw that in the few programs that estimated this competitiveness, it was actually higher than 0.2, which is considered a threshold for cost effectiveness. So being Albopictus or aegypti, we have now some field data showing that it's very much possible to produce irradiated males that will be competitive. And it's interesting to see that most of the genetic programs are not measuring this competitiveness, apart from the Riddle technology, and it will be very important to advise them to do so. So just to give you a picture of what is happening at the world scale now, we have around 34 projects that are starting their baseline collection or even their suppression trials. They are either SIT alone or a combination of SIT and IIT, which is the incompatible insect technology. And 3 of them have reached the pre-operational phase in Mexico, China, and Singapore. And we developed a phase continual approach where we indicated a milestone that should be obtained at each phase before reaching the next one. And in the projects that are funded by IAEA, in the technical cooperation, we expect the countries to fulfill all these milestones before reaching the next phase. And one of the challenges that we saw in the field is that sometimes when people are selecting their field sites, they have problems with the number of sterile males that they can produce regarding the area that they selected. Most of the time, they are not able to select completely isolated areas, and they have some immigration of wild females from the outside, like what was observed with the Bouyer trials in the US recently. And if you take an area of only 200 meters of array, which is around 12.5 hectares, if you want to protect this area against immigrating females, you will have to release your sterile males in another circle of 200 meters, which will require much more sterile males, because you will need 251,000 per week instead of only 62,000 for the central area. And this if you have a target of 5,000 males per hectare per week. So how can we reduce this number? We have different possibilities. The most simple is to select an island. And that is what was done in China. You probably all know this paper that was published in 2019, where the authors report successful suppression in 2 islands of the Pearl River in Guangzhou. More recently, there is a trial ongoing in the US, in Florida, in Captiva Island, which is around 230 hectares, and that you see here on my screen. And the interesting thing with this island is that it's the first one where the technology was demonstrated against screwworms. So it's a reference site. And again, there is a very nice project ongoing, and there are excellent results for the suppression of these populations of Aedes aegypti on this island. The last example of island setting is the one where I'm working here in Reunion Island. And it's funny because it's a ravine, which is a temporary river, which is in fact isolated not by natural barriers, but by Aedes albopictus, because I'm targeting Aedes aegypti in this river, and it's totally isolated from the other population because Aedes albopictus has become predominant, and there is a biological control of Aedes aegypti by Aedes albopictus that is isolating a population. So I will try to eradicate this population in the next month within my ERC project. Another possibility, if you cannot find an isolated island, is to cut your pie. So I have a very nice example in Mexico, where the program changed their release site to select the extremity of the peninsula. And the nice thing is that it will allow them to reduce the number of sterile males that they will need to do their pilot. And they already have excellent results combining auto-dissemination station and release of sterile males. My last slide is to say that we also tried to work on the possibility to test the impact of the sterile insect technique against vector-borne disease. And here it's a big challenge, because what I explained for one small circle of 200 meters ray is true for each of these clusters that you see here with these red boxes. So if you want to do a randomized control trial, it means that for each of your cluster, you will need to make a buffer and release sterile male around. And when I did calculation to do that in Thailand or in Cuba, it means that you would need 20 million of sterile males weekly to do your trial. So we developed a strategy where we will start from a place with a natural barrier, like a sea border, and we will use different technologies to suppress the population. And in the middle of your area, you will have a site that will be cleared from the mosquitoes or with a very low prevalence. And you will maintain this low prevalence by releasing 10% of the number of sterile males that you needed to do the suppression itself. And with this epidemiological setting that we call the wave-based, and it's just a derivation from a step-wage approach, we could demonstrate that you can produce much less mosquitoes than in a standard randomized control trials. The problem is that you will not need more clusters, because all your clusters will change from control to treated during your trial, which means that to have the same power, the same statistical power, you will need more clusters. And so for epidemiological indicators, like diagnostic tests, it will cost a bit more, but you will earn it on your cost of production of the sterile males. Thank you very much.
Thanks a lot. Thanks a lot, Jeremy and Kostas. Really interesting and informative. I want to encourage the audience to go ahead and ask your questions using the chat function. We have a few already, but I want to let people have a chance to enter their questions. Kostas and Jeremy, maybe this is to Kostas primarily, at least this one, but perhaps to both. I'm just going to ask a question of my own here while we're waiting for the audience to chime in. I know that you and others have explored genetic engineering approaches to address, some of the challenges that are associated with getting an SIT program functional. Sex separation being a big challenge for some insects. And I know that the current SIT programs that you're involved with don't rely on genetically modified insects. And I just wanted to know if you could say something about that. And for example, is there resistance among member states to go that particular route, or are there other factors that you've encountered that might make that route more difficult, and one that you perhaps haven't chosen?
Yeah, Dave. This is a question that we receive, quite often. We know that, some member states, are very skeptical, in using transgenic, strains or transgenic products, in general. Yeah. This is why, we at the agency, we are trying and we're focusing on using Mendelian genetics in order to develop genetic second strains which are not transgenic, and make them available to member states which are ready to use them. So there are so many different genetic control methods right now available all over the globe, and it's up to member states to decide which one they would like to use. Yeah, I mean, I'm not sure what you meant by if there are any other reasons. No, I don't. Personally, I don't think there are any other reasons. There is, in several countries there is resistance, by from the public, to some of these technology and products. Yeah, and what we are trying to do, in our laboratories, to provide alternative, strains and products, and and it's up to member states to decide which one to use.
Sure. Great, thanks.
And,
so
yeah, I'm sorry, Dave, and and also I have to say that there is skepticism and concerns not only about the genetic engineering, strains and transgenic strains or other, genetically modified strains. But in the case of mosquitoes, there is also skepticism and concerns in using simply wild-type strains from different regions and countries. And the reason for that is that there are concerns that if there is a production of an X strain in the X country, and this strain is shipped to address a problem in the Y country, and it happened that you have a release of fertile mosquitoes, then and that population is more competent in transmitting the pathogen. That's a concern for the countries.
Yeah, sure.
Yeah, and this is why we decided in the case of the classical genetics sexing strain is to transfer it in different genomic backgrounds and see if the sexing properties are maintained, and in order to address these concerns.
Yeah, great. Yeah, thanks. So I'm gonna go through some of the questions that we have from our audience here. Here's one. Are there cases where the SIT has failed to control the target pest? That's the question.
Maybe if you let me reply, Kostas.
Yeah.
Go ahead.
Yeah, to either of you, I guess. Sorry, I didn't address that to any of you in particular, but yeah.
You can go ahead, Jeremy, and then I will chip in if needed.
Okay, so just to say that, of course, there were thousands of examples of SIT programs that failed. And one of the most impressive is the SIT program against Copioma omnivorax, so the screwworm in South America, because it worked in North and Central America, but it failed in South America. Because we must say that SIT is technology intensive, and when you start to release your sterile males, you must release them all over the time, and you must not stop. If you have any management issue, budget issue, or any other problem that you don't succeed to control, then you fail. And we had several examples. And I started to work on SIT on tsetse flies in Africa. And in this field, there are more examples of failures and successes. And the main success was in Zanzibar. And recently, we expect a successful eradication in Senegal. But apart from that, most of the projects were not sustainable, and we have many failures. And actually, there is a full chapter on the reason why we had failures in these programs in the new SIT book that was published recently, so that we can share it with the participants in the event.
Great.
Yeah, if I can add, Dave,
Sure.
One thing we need to make clear, it's not a failure of the technique. The technique works, and this is what Jeremy tried to emphasize, but it's so complex and managerial intensive and demanding. And most of the failures, are due to these reasons and not because of the technique. Yeah.
It's really requires a strong commitment to keep it going. Yep. Here's a question that's related to something you already said, but it does extend a little bit. You mentioned one of the biosafety concerns relating to genetic background. The question was, what was that concern? I think you addressed that in part, but maybe you could just say it again. But the other part of the question is, were there other biosafety concerns that you had to deal with, such as the consequences of eradicating Aedes aegypti populations themselves? Was that an issue, or any other issues?
Right. I mean, we are not trying to eradicate, the species, globally. Mm-hmm. What we are trying to do with this SIT is to locally eradicate, the species, and or to suppress. No, actually, I have to take it back. What we are trying to do is to suppress the population below the threshold required for disease transmission, and eliminate it from the areas where it was not there in first place. Where we have invasive, mm-hmm. Inversion of the species. For example, Aedes albopictus. Has been, really invasive, and has been spreading all over the globe, and there, you can eradicate it, because it was not there first place. Yeah. So, but the goal is not to eradicate the species, not at all. Mm-hmm. I don't know if you want to add anything, Jeremy, on this.
Good. Okay, so here's another question. How often do you need to release the SIT males over? So how often did you need to release the SIT males over the 6 to 9 week period until the natural population suppressed? And how long does it take for the wild type population to rebound after releases are stopped?
Yeah, I'm sorry if this was not clear during my presentation. These were lab populations, mm-hmm. laboratory cages. Yeah, yep. It was not in the field. So, and perhaps I missed to mention that, the releases were, on a weekly or bi-weekly basis. Yeah. So in the field, perhaps Jeremy may expand on this. So it depends on the destination population and the production and the competitiveness and the longevity, how frequently, sterile males need to be released. Mm-hmm. But, after you suppress or locally eliminate the population, if you don't protect the region by doing preventive releases, for example, and then, mosquitoes can fly or can move, with humans and commodities, and then they can start slowly rebounding and re-establishing themselves in that region. But perhaps Jeremy may wish to say a few things more there.
Just to add that for the field trials that are currently conducted, you have examples between 1 release per week to a 3 releases per week, for example, in China. And in fact, when you start your program, you do mark release recapture to calculate the survival of your sterile males, and then you can make calculations to see what is the most appropriate frequency for your program.
And one more thing, Dave, just to clarify for the audience, mm-hmm. Is that, the same is true for any genetic control method. I mean, no matter if you use SIT or IIT or, the riddle or anything, else, I'm talking about population suppression method. If you stop the intervention and, mm-hmm. You don't do any preventive releases, then, mosquitoes fly and they can migrate and they can invade again the area.
Yeah, definitely not unique. So, okay, great. So here's a question. Starts out with a compliment, and then the question. The compliment is a great presentation, and the animated video was really cool. The question is, do you have any issues with viruses that are often prevalent in mass rearing situations? So, a technical question about mass rearing.
So it's a concern, and we have been developing tests to screen colonies. So of course, we have a lot of biosafety precautions that we take when we initiate colonies. And then thereafter, when we upscale these colonies, they are also protected. And we developed tests that can allow to test your colonies at the beginning. And then if you take biosecurity measures, you can avoid contamination. And it can be not only by arboviruses transmitted to humans, which is a biosafety risk, but also by viruses that are specific to mosquitoes, like densovirus, that can ruin your productivity. And of course, we want to avoid that. So, of course, we have a range of biosafety measures that we take to avoid this problem, but it's a real concern.
Yep. Great. Great. Thanks. Here's a comment, just a comment from Hamadou, who says, just a comment to congratulate Jeremy and Kostas on the nice work and the presentation. So, okay. And the next question we have here has to do with cost. How costly is this program? You know, from a financial point of view, or from a financial standpoint, is it accessible to low-income countries? That can go to either one of you.
Please,
would you like to go? Okay.
I guess the difficulty of the question, the difficulty of the question is already present here. We can see the real.
No, it's not a difficult question.
Okay.
You know, I mean, all population suppression programs require, you know, the basic infrastructure, and the basic infrastructure is, you know, the mass rearing, yeah, is the handling, is the chilling, is the releasing, is the monitoring after, you know, during and after, you know, the intervention, yeah. The only difference is, you know, for SIT is, you know, the means of sterilization, yeah. And in that case, you know, we are using ionizing radiation, and you need an irradiator. Once upon a time, that was, you know, a little bit expensive, yeah, for the gamma cells, you know, then it was a bit costly, and low-income countries, you know, may have not, you know, might not have, you know, the means of doing this. But nowadays, we have tested and validated X-ray machines, and we see that, you know, we can use them quite efficiently, at least at the small scale trials, you know, that are currently being, you know, carried out in different places, shown by Jeremy. And these machines are not that expensive, are similar to the ones that one can find, you know, to her dentist, or in a medical center, or in the hospital. Yeah. So that's the only difference. Jeremy, you may want to add something on this.
Okay. Great. Thanks. Here's a question to either of you. Are there any international restrictions related to radiation? Are there any international restrictions for using radiation devices to irradiate the males?
I think this is.
Sorry. Go on, Kostas.
Go ahead, Jeremy.
No, you know, radio safety is a wide field, and there are of course limitations, and but it's also true for all other applications like radiography, irradiation of food, of blood that you inject to people. So you have many restrictions, but it's one of the areas where the IAEA can help the countries to develop, you know, their human capacity to handle irradiators, and there is nothing that is unfeasible, but of course you need to take a lot of precautions to do it in the proper way, and to protect the people that are using the irradiation systems. Yeah,
there's another.
Again,
yeah, go ahead. Sure.
Go ahead. But again, you know, the fact that we are, you know, we have been testing and validated, you know, X-ray machines, you know, lift some of the major restrictions which apply in other irradiators. Yeah. So an X-ray machine is, as I said, you know, similar to the one that you can, you know, go to your dentist and get you know, an X-ray, or, you know, to a medical center for your lungs or whatever else. Yeah. So they are safe, and you know, they don't have such restrictions that you know, apply for other irradiators.
Yep. There's another question that's related to radiation that I'll ask now, and it has to do. The question is: do you need the radiation sources in the field for irradiation immediately before the release? So I guess it's a question of when the radiation takes place in the process.
Yeah, so there is no need to have it in the field at the place where you release. We have developed mass transport systems. When you keep your males at 10 degrees, and in this compacted form that I showed on my slide, you can transport them for 24 hours without any impact on the quality. And recently we had a trial in Greece that was fully developed with sterile males that were produced in Italy and shipped by DHL. So you don't need necessarily an irradiator in the field. Now, if you want to go for an operational project where you will need millions of sterile males, probably you will need a factory that is not too far from the field. Otherwise, you will lose money with the transportation cost. But if you take fruit flies, for example, hundreds of millions of sterile males of fruit flies are shipped every week in the world. And for example, most of the programs in the US are conducted with fruit flies that are produced in Guatemala.
Yep. Yeah, good point. So I just want to make an observation that maybe for the audience's benefit, that the International Atomic Energy Agency, for whom you work, is involved in this area of pest control or insects, because it's a peaceful application of radiation, and that was one of the motivations for the International Atomic Energy Agency. So that's the connection between SIT and the IAEA. So the next question has to do with what's known about the sexual selection system in the natural populations of mosquitoes. In other insects, there are plenty of documentation which show that even single mutations can have a significant effect on sexual selection systems of the lab-reared strain. Has your mating competitiveness tests been conducted against the natural population?
Yes, on the graph that I presented, it was only field estimations of the competitiveness. So we also have many others in lab populations. But I would like to say something on this, is that the beauty of SIT is that you don't release one mutation, you release millions of mutations. And each individual in your batch has different mutations from the others. So you introduce your mutations at a very low frequency, but you introduce a batch of mutation in each individual, and it's different between individuals. So by construction, there cannot be resistance to this system.
Mm-hmm. Yep. So Jeremy, here's another question to you from someone in the audience. At the end of your presentation, you mentioned a need for 20 million irradiated mosquitoes. Is this an annual production number, or is it necessary to cover a certain area?
No, it was just to conduct randomized control trials, and you needed 20 million sterile males per week. So, and we had a very similar estimation in Havana in Cuba than in Bangkok in Thailand, because finally the incidence of dengue were quite similar, and the cluster of similar size, and the populations of Aedes as well. So we calculated the amounts of sterile males that needed to be released per week, and it was around 20 million sterile males weekly.
Yeah. Okay. Good. Well, here's an interesting question that it may be a little premature for your program, but we'll ask it nonetheless. Is there any data yet that show how well SIT reduces disease incidence? So is there any epi data that?
Yeah, so the only data that I know, okay, first I must say that in China, they had one indicator that is not directly the disease, but they looked at the number of bites per people, during temporary exposures to bites, and it was reduced by more than 99%. So of course, if you are not bitten, you cannot get the disease. But more recently, there were several press releases from Singapore, because they demonstrated that they reduced by more than 90% the incidence of dengue in the central area of their release sites. But it's the combination between SIT and IIT. So, Wolbachia to induce sterility, and irradiation also to induce sterility, but to secure the females that you release. Because if they are sterile, they cannot establish in the population, and so this combination has many different advantages.
Mm-hmm. Mm-hmm. Yeah, great. Okay, I have another question here for you that has to do with the impact of SIT, and I think the question here is what's the impact on pollination in certain regions? So I think the question is looking for the questioner is sort of asking about ecological, environmental impacts of these programs.
So we have a lot of data for pests. We don't have any operational program for mosquitoes now, so we don't have the data for mosquitoes. But for example, recently we published the result of a study of almost 10 years of monitoring of the non-target fauna in Senegal in a Tsetse SIT project. And you know, by the SIT is species specific, so in itself it has no impact on the other species. But the problem is that generally we have to integrate it with other control measures, specifically insecticide, that can themselves have an impact on non-target fauna. But the good thing is that when you look at the SIT program in Senegal, it targeted elimination. So you need to use your insecticide for a certain time window, which is 1 or 2 years, and then it's fully replaced by the release of sterile males. And so you have a minimal impact on the environment. And in that project, we demonstrated that there was a very small impact that was also very short in time, 1 or 2 years, and then the biodiversity restored to the same level than before the program.
Mm-hmm. Great. So got a couple more questions here, so I'm going to keep going. So the comment first is, hello, thanks for very interesting talks. And if I understand, the question is, if I understood correctly, unless I missed it, the Albi strain with the inversion had a better flying ability. Is there a reason for this? And furthermore, it appears that the strain with the inversion suppresses the cage population more slowly, less efficiently. Does it mean that the flying ability is not very relevant, or is the difference too little to make a judgment?
The strain is for the aegypti, both with and without the inversion. And the comparison was between the GSS with the inversion with the wild type strain. But we saw that the red dye strain with and without the inversion are better flyers than the wild type strain. So it's not that it was, that there's a specific reason, doing inversion. I think it's an intrinsic property of the red dye mutation. I don't know why. It seems to be more fit and has better rearing efficiency and better flying ability, at least against the wild type strains that we have tested with. And we have done this against the Brazilian population, wild type population, and Mexican wild type population. So it has nothing to do with the inversion. It's probably related with the genetic characteristics and of the red dye strain itself. Now, the second one, I think it's right. It's, the difference between 6 and 9 weeks and the laboratory case population. I wouldn't like to make any, any strong conclusions out of it. The differences might be too little, to be judged at this point.
Mm-hmm. Mm-hmm. Yeah, so we're coming to the end of our questions. I have a couple on my own notepad here that I'd like to ask you. In the past, you guys have worked on Anopheles arabiensis, and I saw some data actually on Anopheles arabiensis in your slides, and possible trials of SIT in Africa. And I just want to ask where that work stands and how that particular application of SIT is going. Could be either to you, Kostas, or Jeremy.
As you like.
Go ahead, Jeremy.
No, I was just going to mention that we have currently a pilot trial that will start soon in South Africa. We are collaborating with this country to develop SIT against Anopheles arabiensis. in an area where they did not succeed to reduce the malaria below a certain threshold, because they saw that most of the transmission was occurring outside. And so they decided to try and reduce the wild populations of Anopheles arabiensis to eliminate malaria. And one interesting thing in this project is that the sexy system towards which we are going now is one based on fluorescence of the males. The males are fluorescent and the females are wild. And if this strain is selected for the field trials, it will be a first case where SIT will be combined to a transgenic strain. And it will be very funny because we are trying to see what it will mean in terms of regulation. Because as you know, transgenics are very much regulated, but the advantage of irradiation is that it confers sterility, and an organism that is sterile is not regulated in the same way than an organism that is fertile.
Yep. And I missed where that field trial was taking place. Where was that?
South Africa.
South Africa. Yeah. Okay, good. So you have a number of trials that are ongoing in a variety of places. In fact, it was a fairly impressive number when you showed the map. How easy or hard is it for you to establish trial sites and to do that? Could you talk a little bit about that trial, establishing trials for this technology?
Yeah. First, I want to say that at IAA, we don't select field sites or establish trials. We help member states that want to do so, so we are here to help. And
so they solicit.
So you have, it's under a request, so we don't do the work for them. It's their choice, and we help as best as we can, but it's always a country decision. And so, as you could see now, we have a lot of requests from different countries. But the interesting thing is with our work is that we participate to all these programs, and we have the opportunity to see progresses on different aspects of the project in different countries. And the role of the IAEA is that we try to take the best part of each of these projects and to share it with the other member states. And it allows, pushing the technology globally and going faster to have good results, because you earn a lot of time.
Yep. Well, we're just about at, speaking of time, we're just about out of it. I'm going to stop the questions now. We've sort of come to the end of them. I want to thank both Jeremy and Kostas for a great presentation, really interesting and stimulating. The questions were reflective of people's interest in what you're doing. It's really, really, really interesting. And thanks again for joining us today. Before we sign off today, I did want to mention to the audience that we have one more series, one more webinar in this particular series on genetic biocontrol. That'll be held the same time and place next week. It'll be on Wolbachia and a technique called the incompatible insect technique, using Wolbachia as pesticides against mosquito populations. And this will be given by Dr. Stephen Dobson, who has been a real innovator and developer of this technology, and a collaborator from Verily Life Sciences, Joe Ohm, will be a co-presenter. So I encourage everybody to come and hear about that. It's a technique that's related to SIT. In fact, is being used in combination with SIT. And I think Jeremy and Kostas mentioned that trial in China, which was in fact a combination IIT-SIT program. So with that, Jeremy and Kostas, thanks again.
Thank you, David.
You're welcome. Thank you, David, for the invitation.
Yes, and thank you for the audience. And with that, I'll say good morning and good afternoon, and we'll see you next week.
Thank you.